Although the DNA in each cell is different, is the genetic information stored within it the same? All cells contain the same genetic information in their DNA, but gene expression varies based on cell type, allowing cells to perform different functions. The DNA in cells is structured as a double helix, with one strand containing adenine, A, thymine, T, cytosine, C, and guanine, G, bases, and the other strand having complementary bases. This complementarity allows DNA to be replicated and transcribed. During cell division, DNA is replicated into two strands, with each strand moving into the nucleus of the new cell. Upon reaching the new cell's nucleus, DNA binds with proteins that regulate gene expression. These proteins determine whether specific genes are expressed or suppressed. For example, brain cells and muscle cells share the same DNA, but brain cells express genes for neurotransmitter production, while muscle cells express genes for muscle protein production. Therefore, while all cells share the same genetic information, gene expression varies by cell type, allowing cells to perform different functions. Although the DNA in each cell is generally the same, there may be slight differences in the nucleotide sequence. These differences can occur during DNA replication, where errors may lead to minor changes in the sequence. Such errors can happen naturally or be induced by exposure to radiation or chemicals. Small variations in DNA base sequences can significantly impact cell function. For instance, changes in gene sequences may lead to incorrect protein synthesis, potentially causing diseases or functional disorders. Here are examples of DNA differences in different cells. 1. Cell type differences. Brain cells and muscle cells share the same DNA but may have slight differences in nucleotide sequences, contributing to functional distinctions. 2. Individual differences. Within the same species, different individuals may have slight variations in DNA sequences, leading to individual differences. For humans, these differences can affect traits such as height, hair color, and eye color. 3. Disease-related differences. Genes associated with diseases often exhibit variations in nucleotide sequences. For example, genes linked to breast cancer may have single nucleotide differences. Therefore, while DNA in each cell is generally the same, slight differences in nucleotide sequences can exist. These differences play a crucial role in determining cell function and contribute to the complexity of individual and cellular diversity. Do cells repeat the process of creation, replication, and extinction? So, when cloned, is the genetic information the same? Yes, cells undergo a repetitive cycle of creation, replication, and demise. In the creation phase, cells divide to generate new cells. Cell division consists of two stages: nuclear division and cytoplasmic division. During nuclear division, DNA is replicated and moves into the nucleus of the new cell. DNA replication involves the duplication of a single DNA strand into two complementary strands, allowing each strand to move into the nucleus of the new cell. In the cytoplasmic division stage, the cell's cytoplasm divides into two new cells through processes like parallel division, intermediate division, or vertical division. In the replication phase, after the formation of a new cell, DNA can undergo replication again. DNA replication is essential for the growth and division of cells. In the demise phase, cells die. Cells can die due to aging, damage, diseases, and other factors. While genetic information is generally maintained during replication, errors can occur in the DNA replication process. These errors may arise naturally or due to exposure to radiation or chemicals. Small differences in DNA base sequences can have a significant impact on cell function. For instance, changes in gene sequences may lead to the incorrect synthesis of proteins, potentially causing diseases or functional disorders. Therefore, cells repetitively undergo the processes of creation, replication, and demise. While genetic information is usually maintained during replication, errors can occur.